Good evening, everyone, and thank you so much for coming out in such numbers. It's really amazing to see so many faces. 60,000 years ago, humans like us weren't the only humans on the planet. There were several others. Homo floresiensis, for example, a diminutive 1.1 meter high hominin that lived only on the island of Flores. This species was only discovered in 2005. We don't know what happened to Homo floresiensis. There's been suggestions that Flores man or Flores person is still with us today, but lurking in the jungles of Flores, but no sightings have been made recently. We don't know quite when he disappeared, but recent dating evidence suggests that it's more than 50,000 years ago. Neanderthals, of course, the archetypal cave person. Neanderthals have evoked all sorts of fiction, movies, books, and the story of Neanderthals and who they were and where they lived and how they disappeared is something that a huge number of people have done a huge amount of work on, and I'm going to be talking about aspects of that tonight. Anatomically modern humans, of course, like us. As the story will unfold tonight, you'll hear that we evolved in Africa and then moved out. And a lot of debate is undergoing at the moment regarding how this process worked, when we moved out of Africa, how many times, and when. And finally, a mysterious new hominin that appeared on the scene in 2010, Denisovans. We don't have a face for Denisovans yet because the fossil record is very silent on them. We only have one small finger bone and 3 teeth at the moment of the Denisovans. So we have to have a lot more fossil evidence before we can put a face and reconstruct what we think these people look like. Now tonight I'm going to talk about Neanderthals, modern humans, and Denisovans, and what happened between 60,000 and 30,000 years ago. This crucial period where we go from a number of hominins to just one. Firstly, I'm going to talk about early African origins, the long duration, the long story of the fossil record and how we came to be part of it. And then I'm going to talk about Neanderthals, physiologically who they were, where they lived, what date they disappeared, and our relationship to them. And then I'm going to move to discussing our own species, anatomically modern humans, and the story of how we came out of Africa into the rest of Eurasia. I'm going to talk a little bit about the genetics of the process, because genetics is hugely important, obviously, at the moment, and the genome, the data that we've got, is really shedding light on exactly who these people were and what they did and where they lived. And then finally, I'm going to talk about Denisovans and some of the most recent work. This is a really fast-moving field, so I'm hoping that I can give you the recent and the most up-to-date data and up-to-date information that's coming out regarding all of these issues. Okay, let's deal firstly with early African origins. This is a family tree of humans, going from 6 million at the bottom to the modern day, the period at the top. So it's an extremely long process. We know that we shared a common ancestor with the ancestral chimpanzees and other great apes about 6 million years ago, and since then our family trees have diverged. In this illustration, you can see that everything in blue is the genus Homo, and everything in red and different colors are off our supposed evolutionary line. The tree of human life has become extremely bushy over the last few decades. It used to be so simple when I was an undergraduate. I mean, people just evolved one into another, and then eventually there we were. But now it's become extremely complicated. To make things really simple, however, I'm just going to focus on the last 250,000 years, and mostly the last 60,000 years. When you see at the top of this image, the skull of a Neanderthal and the skull of a modern person. But that's just really to place you in some kind of a perspective. Now, in 1987, there was a really important paper published. It wasn't in Newsweek, that was just a popular article. It was published in Nature, the journal Nature. And it was published by Alan Wilson, of course, the great biologist and molecular evolutionist from this country, along with 2 very bright graduate students, Becky Cann, who was the first author of the paper, and Mark Stoneking. And what they did was they took 147 modern humans and they analyzed their mitochondrial DNA. People came from right across the globe, Africa all the way to Australasia. And what they found was that in Africa there was a greater diversity in the mitochondrial DNA than there was outside. So everybody outside has less diversity. And this is consistent with a model that suggests that a small subpopulation of Africans moved out of Africa and populated the rest of the world. And this is the reason for this disparity in the diversity. And this model which is variously being called Out of Africa, the recent Out of Africa, the recent African model, and mentioning, of course, the mitochondrial Eve, has focused upon this idea that ultimately every modern person is an African originally, and that is the cradle of humankind. It's very interesting to, of course, remember that Charles Darwin predicted this. Darwin predicted that Africa would be the source for the evolution of humanity. And this was based on the common ancestral idea, and he was dead right. And time again, we find that Africa is the engine room for evolution of the Homo genus. Quickly moving on then, because I've got a lot more to talk about, I want to talk about Neanderthals. Neanderthals are really cool. These are two reconstructions that were done a few years ago by two sculptors called the Kennis brothers, and they're Dutch, and they were asked to recreate a modern human and a Neanderthal for a big exhibition that took place as part of a project that I was involved in called the Ancient Human Occupation of Britain project. And on the left-hand side here is their version of a Neanderthal, and here on the right is an anatomically modern human. There are some quite significant differences to look at. First of all, in terms of the skull, Neanderthals had quite a long, shaped skull, quite big. They're, on average, a bigger brain size than us, around 1,600 cc's compared to modern humans. But of course, bigger brains don't necessarily mean clever people. They also had some features of the skull which are markedly different to ours. So they had quite a large supraorbital torus, the brow ridge across here, which is often associated with dissipating the muscular forces that are generated when humans chew things. Their nasal passages were quite large and flared, and this is often associated and often explained by the idea that these people were living in Ice Age Eurasia, so very much colder conditions than anatomically modern humans. And this may be an adaptation to dealing with large amounts of cold air that they had to breathe. Below the neck, there were some differences as well. If you, for example, took a femur of a Neanderthal and cut it in half and looked at the cross-section and compared it with a modern human, you'd find that Neanderthal human bones are much, much thicker, much more robust. And again, this is likely an adaptation to colder conditions. Shorter, squatter, more muscular. They also had some other curious features like a V-shaped rib cage. We'll come back to that later on when we talk about the DNA. Anatomically modern humans, on the other hand, taller, less robust, more willowy, likely reflecting their adaptation to living in tropical environments in Africa, where ultimately, of course, around 200 to 180,000 years ago, our species evolved and originated from. The distribution of Neanderthals is interesting. This is a map that shows the locations where we find Neanderthals living. And this is based on the presence of their fossils and also the archaeological evidence that we link to them. And so you can see that they're concentrated mostly in Europe. They go down into the Near East, down in here in Israel, modern-day Israel, into Iraq, Iran, and since 2008, we now know that they also were present in the Siberian Altai because some geneticists in Leipzig analyzed the DNA of a large femur from a robust individual, and the DNA suggested it was Neanderthal. So we pushed out the distribution all the way out here. Neanderthals may have been more widely distributed even than this, but this is where we're fairly sure that they lived. They could, for example, have also lived down in Saudi Arabia and perhaps in the subcontinent. But at the moment, the evidence is hard to interpret. One of the big questions about Neanderthals is when did they go extinct? The why they'll go extinct, we may get onto in questions, but when did they disappear is something that my team in Oxford has been really working hard at and trying to understand. And it all revolves around the reliability of dating. So this illustration was taken from a Nature Review article in 2006, and it shows the distribution of very late-dated Neanderthals. So the ones here in the south of Spain, at a series of sites, including Gorham's Cave and Gibraltar, are generally associated as being examples of where we find Neanderthals that are less than 30,000 years of age. And this site here at Mezhmyskaya in Russia is another one where we find Neanderthals dating to, according to this illustration anyway, between 30 to 35,000. So there are some quite late surviving Neanderthals, even less than sometimes 30,000 years of age. Now, my expertise is in dating and archaeological dating using radiocarbon. Radiocarbon dating is a fantastic technique, but when you get down to 30 to 50,000 years ago, you're dealing with much, much smaller amounts of radiocarbon than you are in the present day. So For those of you that know a little bit about it, radiocarbon has a half-life of 5,568 years. That means that every 5,568 years, half the radioactive carbon in the bone or the charcoal that we find in an archaeological site has decayed back to its parent isotope, nitrogen-14. So basically, 10 half-lives is the limit of any radioisotopic dating method. So when you get down to 30,000 years ago, you've got about 3% of the amount of radiocarbon that you've got in the present day. Just 3%. When you get down to 50,000 years ago, you've got 0.1%. That's a tiny amount. As a consequence of this, small amounts of contaminating carbon can really affect the reliability of the dates unless they're removed. So in the case of a bone, for example, that is 50,000 years of age, if you have 1% contamination with modern-day radiocarbon, you'll produce a date that's over 7,000 years too young. And so this experience of ours, and knowing about the contamination issues, led us to be somewhat skeptical about some of these radiocarbon dates and these estimates here, because we were worried that some of them could well be underestimates. So what we decided to do was to try and address these issues by a new program of dating. This is a particle accelerator. It's the radiocarbon accelerator in Oxford, where I work. And it's a 2.5 million pound piece of kit that enables you to date really small pieces of bone, really small pieces of charcoal. We're talking here about dating about 1 milligram of carbon. That's really a tiny amount. And as a result of this extremely small size requirement, we're able to date things using much better pre-treatment chemistry designed to remove the contamination that otherwise would affect the reliability of our dates. And so over the last few years, we've been working quite hard on improving this pre-treatment chemistry. So for example, what we do is we take a bone like this, we drill the sample with a dental drill, get about half a teaspoon size aliquot of bone, and then we extract the collagen from the bone. And this is this little stuff here that you can see on the end of this set of tweezers. It looks a bit like cotton wool. This is the protein fraction of your bone. About 20% of your bone is protein, and the rest of it is a mineral cement. And the protein is the best material that we can get for dating. So what we do in Oxford is we use something called an ultrafilter, and I'll just zoom in on this so you can see it. This little ultrafilter, we put the proteins into this filter. They're in a liquid form, and then we zoom them around in a centrifuge, and the centrifuge and the filter together act to remove small molecular weight contaminants that come out of the collagen. We trap most of the collagen above the filter, and this is what we end up radiocarbon dating. And we started using this in 2001, and we noticed immediately some big differences between dates previous to ultrafiltration and post-ultrafiltration. With the ultrafilter, we started to get dates that were a lot older when we redated samples that we thought were a little bit too young. And so this ultrafiltration technique is something that we've used a lot in our radiocarbon dating of Neanderthal age material. And I want to give you a quick example of some of the results that we've got, because what we decided to do is to go back to some of these sites and test them again. In some cases, it's a little bit difficult to do this because curators don't really want you to go and drill their precious fossils anymore. But you know, when you're sort of persuasive like me, you sometimes can do it. So here's an example of the kind of effects we're talking about. This is that site in the Russian Caucasus called Mezhmorskaya. And they found there 2 amazing little infant burials, Neanderthals, small dead Neanderthals, complete remains. And in a few years ago, 20 or so years ago, one of these bones, one of these skeletons was dated to just over 29,000 radiocarbon years BP. So we went back to the same collection of material and we dated another Neanderthal skeleton which is found above this, so it therefore should be younger, and we got a date which was much older, around 40,000 years ago. And this to us indicates another in the long pattern of underestimated radiocarbon dates based on insufficient decontamination. Another one we've got, another site in the south of Spain, very, very young dates here. This date here of a bone found near this amazing complete mandible with a date of 33,000. We redated the same bone. These are just 2 of the dates. We've done the same date about 4 times. And they're always greater than ages. That means they're beyond the radiocarbon limit, more than 48,000 years ago. So, one by one, we've managed to go through these late surviving supposed Neanderthals and remove them from the record. Not only that, though, we've also gone back and we've done a huge amount of dating from other sites, the really important sites in Western Europe. We've dated around 40 sites from right across Europe, all the way from the west of Spain over to the Levant and to Russia. And these are the summary data of all of the results we've got. So what you're seeing here are dates of the most recent archaeological sites which contain Neanderthals. And what you can see here is that they all cluster to between 45 to 50,000 years ago. And we can calculate a more precise date for the end or the demise of Neanderthals using a statistical technique called Bayesian modeling. And basically what we get is a range between 41,000 to just over 39,000 years ago. That is our estimate of the extinction of Neanderthals in Europe, based on 40 sites and more than 200 radiocarbon dates. And this is a summary of about 7 years of work. And it was published, this paper, last year in the journal Nature. And we found, as I say, we found this result on the basis of all of this work. So that gives you a little insight into the disappearance dates of Neanderthals. Now I want to move to looking at modern humans. So as I said at the beginning of the talk, modern people like us originated in Africa about 200,000 years ago. And a lot of archaeological work has been done to try and understand when our species moved out of Africa into the rest of the old world. Because, of course, we've got a lot of evidence from different parts of the old world for the presence of modern humans like us. And later, the diaspora continues, of course, into North America and South America after around 15,000 years ago, into Australia and Melanesia as early as 45 to 50,000 years ago, and ultimately, of course, right across the Pacific to the very final landmass that was settled by modern humans, and that's New Zealand. So we have evidence from the archaeological record that modern humans got out at least twice. The first is around, we think, 110 to 120,000 years ago, which is evidenced by some archaeological sites in the Near East. There are several sites where we have early anatomically modern humans, but they get replaced by Neanderthals a bit later, so we find Neanderthals coming in later. So it seems to be somewhat of a failed excursion, if you like, of people out of Africa. And then much later, we think around 60,000 years ago, maybe a little bit younger, we get another movement of people, which results in the greater diaspora of people right across the old world. In Europe, there's a huge amount of archaeological evidence that covers this period. And we think that the reason for this is, well, twofold. One is that there are a huge amount of cave sites in Europe which contain the archaeological record of this particular period. And the second is that there's a long record of archaeological excavation spanning more than a century. So we have a lot of excavated sites and a lot of material in museums that has been studied. And there are two main ideas about the movement of modern humans into this period, into this part of the world at this time, between about 45 to 35,000 years ago. The first is the so-called Danube corridor hypothesis, and that's the red arrow that you can see at the top here. And that focuses on the potential movement of people up the Danube corridor, which is this obviously major waterway, major artery. People need water, and so we think that they usually might focus on moving up waterways like the Danube. And there's a lot of archaeological evidence for early anatomically modern humans up this corridor. And one of my PhD students is focusing on this at the moment with her work. And the second suggestion is a sort of southern route, which is the southern Mediterranean route, which involves people moving along this corridor past and through Greece, Italy, and into Europe that way. At the heart of this, again, we rely upon radiocarbon dates to help us to unlock the when and the how this happened. And this is the subject of a lot of ongoing work. And what's really interesting about the arrival of modern humans at this time is the difference that we can see in the archaeological record between Neanderthal material and modern human material. There is a period at around 50 to 60,000 years ago when modern humans and Neanderthals are doing pretty much the same thing. They're making similar tools, which we call a Mousterian toolkit. And you can see this here under this Neanderthal guy here, the types of tools we're talking about. They're mainly flake-based tools like this. So they're flakes cut from a larger core, and they also have characteristic tools like this Mousterian hand axe. And these are the types of tools we see for about 250,000 years in the archaeological record of Europe and the Near East. We also sometimes see Neanderthals using pigments like this. This is red ochre or hematite. And sometimes We find these blocks of pigment at Neanderthal-related sites. We're not quite sure what they're doing with them. They may be using them to self-decorate. They may be using them to draw things. We simply don't know. But that we do know that they were interested in this type of material. Towards the very end of the Neanderthal period on Earth, we find that they're also doing some interesting, more innovative things that seem to be the more common hallmark of modern humans, which you can see here on the right. This includes things like making ornaments out of shells. These are pierced shell ornaments, about 2 centimeters high, a very distinct type of shell called Nassarius. And these Nassarius are found commonly amongst sites of the Aurignacian, these early modern human sites across Europe. We also find a plethora of these tiny bladelets that you can see here at the top of the picture. These are a few centimeters high, and they're very, very finely made. We don't really know what they're for, but the suggestion's been made that they could have been the part of composite points. In other words, they could have been attached to spears, or maybe even bow and arrows, we don't know. These tiny points could have been made into composite implements. They also manufactured large blades as well. But it's the other things that modern humans seem to have been doing in the period of a few thousand years after they arrived into Europe that really captures the imagination of archaeologists and the general public. In the south of France, here in the Ardèche region, we find the arrival of artistic expression. This is material and cave paintings from the site of Grotte Chauvet in the Ardèche region, and you can see here an amazing assortment of material from the Ice Age art. These are representations largely of animals, animal figures, including horses that you can see here, woolly rhinoceroses at the bottom of the picture fighting each other. We've got big cats, we've got hyenas, we've got bears. These big cats are amazing. They have little marks where the whiskers go at the front of the snouts, you see here. This is an identification that modern conservationists use in Africa to identify individual lions. These cave lions, I mean, it's possible, I guess, they could even be individually identified by the modern humans that painted these images, because they each have their own specific whisker patterns that you can then use to identify them in the archaeological in the wild. Amazingly, early anatomically modern humans also made music, and at 2 sites in the south of Germany we have this evidence firsthand in the form of flutes. This is a flute that's made from mammoth ivory. It's carved mammoth ivory, and the holes are punched in, the middle is hollowed out, and it's stuck together. And it makes the same range of sounds as a modern recorder would make. We have flutes made of griffin vulture bone and mammoth ivory from 2 sites, Geisenklostler and Hollefels, that are in the southern part of Germany. And usually these are fragmented. They're not found in one piece like this. They're usually broken up into small bits by virtue of the time they've spent in the archaeological record. And they need to be put together very carefully by archaeologists. In fact, flutes like this have been formed from up to 80 different fragments. So what do these things sound like? Well, shortly after one of these was discovered, the Hohner company in Germany reconstructed one, and it was able to be played. And it plays the same range of notes that a modern recorder would use, as I said. And I was very fortunate because my colleague Nick Conard, who's the excavator of this material, has allowed me to share the sound of one of these things and what it played. So if you listen carefully, in the background to this sound file, you hear the drip, drip, dripping of cave drip as it comes through behind the noise of the flute being played. We don't know the music, of course, but we know the sounds, and this is what it sounds like. We dated this site in 2010, and the levels in which this material was found date to between 38 and 40,000 years ago. It's incredible to think that modern humans, like us, living in caves, were making music together. And because it tells us so much, it's so evocative. When people make music, it transports them to different places, and it turns the people into different things. So it's a really fascinating insight into how people behaved. People at this period also made mobiliary art that they could carry with them perhaps, and they carved things out of ivory. We have the earliest representation of a horse at the site of Vogelherd in the south of Germany. We have pierced ornaments made of deer teeth. We have carvings of mammoths. We have carvings of bears, all sorts of different animals. In 2001, on the German side at Hollefels, this little piece of ivory was found, and it's clearly been carved. And when it was discovered, the archaeologists were interested in knowing what it might be, but they had no positive idea because it wasn't very, very clear. It's hard to identify exactly what it is. And then the following year, they found another piece of worked ivory in about the same place, and they noticed that it was broken, and when it fitted back together, it made a water bird. This is, I think, the earliest representation of a diving bird. It's, as I say, it's about 35 to 36,000 years ago, and it's pretty clear once you stick the head on it what it is. What's the earliest evidence for anatomically modern humans? Well, the earliest definitive evidence comes from this site in Romania called the Peștera cu Oase, which means the cave of the bones. This is a cave fissure that was found by speleologists. Many of these sites, like the Chauvet Cave, are found by speleologists, people hunting and looking inside the caves to see what's there. And these explorers were finding in several of these fissures, they were finding evidence for animal bones. So once you get into this amazing cave site at the Peștera cu Oase, you have to go through several sumps where you dive into water, come along a cavern, come up into a cave, open cave, and then go along it, and then dive into another sump full of water because of the spring that's there. So it's not the kind of thing that I'd really want to do. A little bit dangerous. I'm more of a dry land lab guy. I don't really want to go and sort of scrape around here like this. Claustrophobia, no. Anyway, luckily we didn't have to go and dig around here because these guys found this. And this is the jaw of an anatomically modern human. We can tell that because it's got a chin, and Neanderthals didn't have chins, whereas we do. And this is a sample. This sample was sent to us in Oxford, and we got a date of between 39 to 41,000 years ago. Interesting date, if you'll recall the date of the Neanderthal extinction and demise. At the time, we didn't have precise dates for Neanderthal disappearance. But this is a direct date from this specimen, and it's the earliest directly dated anatomically modern in Western Europe. The person that identified that thought that it had mosaic features between modern humans and Neanderthals, but a lot of people were quite skeptical about that. He saw characteristic traits from Neanderthals as well as from modern humans. Although it was a modern human, he could see something else in it. And his prescient observations came to be very, very significant. In a second, I'll tell you a little bit more about the genetic story. Genetics has really blown this field apart, and we're really, really privileged to be living in this period where there's so much really good science being done. And paleogenomics is a really exciting area of research. So everyone's familiar with DNA testing. DNA testing, hardly a week goes by where there's some evidence for DNA testing coming to the rescue of the legal profession, of the police, in finding murderers. And this is revolving, of course, around modern DNA. But paleo-DNA, ancient DNA, is a completely different field, and one in which it's much, much more challenging because, of course, rather like with the collagen, the amount of DNA in a bone slowly starts to degrade and disappear over time until eventually there's nothing left. In certain parts of the globe, in warmer environments, DNA really doesn't survive at all. It's only in the colder parts, the more temperate locations, that you get a lot of DNA. But with modern techniques, even the more difficult locations and older locations are starting to yield genetic data that can be relied upon. Those of you that remember, or those of you that are still at school, will know a little bit more about how the DNA side of things works. So there are two main types of DNA that we can use in ancient DNA research. They come from two different parts of the cell. So one is the mitochondrial part of the cell, which is here. That's the energy-producing parts of the cell. This is mitochondrial DNA. And here you get short sequences of, 16,000 base pairs in length, which are matrilineally inherited. So they come down from the mother to mother to mother all the way through. And there are lots and lots and lots of them, repeats, but they're short sequences of 16,500 base pairs. On the other side of the coin, in the nucleus, is the nuclear DNA, which is diploid inherited, coming from both parents. And this genome comprises 3.3 billion base pairs. And consequently, it has a great deal more information for us if you want to understand the past, and particularly the phylogenetics, the history of populations. This is Svante Paabo. He's a colleague of ours from the Max Planck in Leipzig, and he has been at the forefront of ancient genetics work in the Paleolithic, ancient DNA, particularly of Neanderthals. And in 2005, his team at the Max Planck and a group of other researchers stated that they were going to attempt to sequence the Neanderthal genome over the next 4 to 5 years. And this very ambitious project was built around developments in instrumentation, which we call next-generation sequencing. Without going into too much of the detail, basically what this means is that you take, where you're able to, small sections of the DNA, and you can knit them together using a fantastic instrument, a fantastic machine made by several manufacturers now, which allows you to build up lots and lots of small pieces of DNA and can compare them to genomes of modern humans that we all, of course, already have. So Svante and his team were looking for bone which had suitable DNA preservation, and they searched quite far and wide, and they eventually found 3 bones from the site at Vendia Cave in Croatia, here they are here, which had quite good DNA preservation. And these 3 bones furnished almost all of the Neanderthal genome DNA data. In the end, in 2010, when they published this paper, the draft sequence of the Neanderthal genome, they'd sequenced around 55% of the Neanderthal genome. And whereas previously we had no evidence and no information from the mitochondrial DNA that there was any interbreeding or any interaction between modern humans and Neanderthals, the genome of the nuclear side of the coin completely changed the interpretation, and it showed clearly that we had interbred with Neanderthals. And it also allowed us to put a figure on the amount of DNA that we share, and that figure is between 1.5 to 2.1%. Outside of Africa, all non-Africans share a very similar amount of this DNA from Neanderthals. So if you're an Australian Aborigine, or a person in France, or a person in China, India, whatever, you have almost exactly the same amount. So this means that the movement of the ancestral populations of these modern humans must have happened after the interbreeding event with Neanderthals took place. So it's a particularly important piece of research. And this enables us to look at some really interesting questions. So, for example, what did we get from Neanderthals? What was it that Neanderthal DNA gave us? So I bet you've all had this thought experiment at one time or another about certain people you know, maybe, I don't know. I mean, it's the Rugby World Cup, right? So flatmates you've had that sort of didn't clean up much after you, I don't know. My wife said, oh, you should put up some All Black pictures. I said, what, are you kidding me? They're anatomically modern. They're not Neanderthals. No. Anyway, we know from the DNA that we actually have some positive inherited genes from Neanderthals. And one of these are genes that affect the keratin. And these groups of genes appear in our genome, and they are directly coming in from Neanderthals. And they seem to give us better adaptations for skin and hair. The quality of the hair is better. And this, intriguingly, could be due to the fact that when we came out of Africa, we were coming from our tropical environment, and we're coming into the Ice Age realms, and it may be that this gave us a better adaptation, and therefore it was positively selected, and perhaps this is why these genes came into us. But on the negative side, we also have some groups of genes that are responsible for several other things. For example, Crohn's disease, diabetes type 2. Diabetes type 2, this group of genes which will give you diabetes or a higher propensity to get diabetes, is linked strongly to Neanderthals. This doesn't mean that Neanderthals had diabetes type 2. In fact, in Neanderthals, it almost certainly had a reverse influence and a reverse effect, because it could be linked with starvation adaptations. These genes are also linked with that. And it also doesn't mean that Neanderthals went around smoking cigarettes either. This simply refers to the fact that there are several genes linked with compulsive, addictive behavior personalities that we also find coming from Neanderthals, as well as several of these other quite negative things. So it could be that in the time since we interbred with Neanderthals, there have been these negative genes that have come into to us, that the evolutionary process of these, as it happens, is currently sifting through and selecting against. Interbreeding, though, is a very, very key part of our understanding of the process of the replacement of Neanderthals by modern humans. And this is what I'd like to turn to now, because the last 12 months have seen huge breakthroughs in this field that I really want to share with you because they're very exciting. So this is a site in Siberia called Ustashim, and it's a site on the banks of the Irtysh River in Siberia. And in 2008, some colleagues of ours, some Russian fossil hunters, discovered an interesting bone, a femur of a modern human. This would only have been found, the discovery was only made possible because other Russian colleagues, paleontologists, knowing that at the moment the permafrost is melting in these areas, releasing large amounts of sediment that contain often the bones and the tusks of extinct mammoths that they can then sell on eBay. I'm serious. They contacted some of these fossil hunters and they said, by the way, look, if you ever find some non-mammoth material that's potentially human, would you be able to just give us a call and then we can maybe study it? And so one of these guys, Nikolai Perestov, succeeded in getting hold of this human bone. And a small piece of it was sent to us in 2010 for dating. And it produced a date, two dates, identical, about 4 years apart. We dated these two samples 4 years apart. And the calibrated ages, the corrected ages for this is between 44 to 46,000 years ago. That's the oldest directly dated modern human outside of Africa. Just a chance to find on the banks of this river. A small piece of this bone was then sent to Leipzig to the geneticists that we work with, to Svante Paavo, and they found exceptionally good preservation of DNA. So good, they were able to get a complete genome. Now, this is a fearsome diagram that I want to share with you. And let me just take the time to explain it because it's really interesting. So basically, these are parts of the genome of modern people. French person, someone from Sardinia, China, South America, Papua New Guinea, Australia. And this is a section of the genome from chromosome 12, so one of the chromosomes. And everywhere you see a colored line, this little yellow or the blue here, yellow, this is Neanderthal-derived DNA. So alleles that expressed coming from Neanderthals. So you can see across each one of these individuals, chromosome 12, we've got one or two little bits and pieces here of the genome. So adding up to around 1.5 to 2% of the genome across all of these sections of the chromosome. Now, if you imagine that you're a modern human and you have an interbreeding event with a Neanderthal, your offspring will have half the genome from them and half from you. And subsequently, that material will be broken up and it'll become recombined into smaller fragments. This fragmentation takes place in successive generations. So the closer we go back to the original interbreeding events, we should expect that these DNA sections that come from Neanderthal should be bigger and more chunky. So what did the DNA from the Ustashim look like? Well, this is it at the top. And you can see here, there's a characteristically quite large chunks of DNA coming from Neanderthals into this modern human person, dating to 46 to 44,000 years ago. Now, it's possible because we have this data and this information, we have lots of genome data now, that it's possible to calculate how long since this integration, since this person lived, how long back was this integration, this gene flow, this interbreeding event from a Neanderthal. And it calculates out to around 330 generations prior to the life of this person, which means that about 50,000 to 60,000 years ago, which is interesting because it's about the same date as we think modern humans come out in this last movement out of Africa. So it's really fascinating data, but there's something else here which is also interesting because there are sections where it's on the other parts of the genome where there are other big pieces. It was also suggested in the paper that there could have been two interbreeding events, one deeper in time and one more recent. Some really interesting information about this came out about 2 months ago in the journal Nature. And this specimen, which hopefully you'll remember from the previous few slides, this is the Pastera-Coase mandible, which we dated, remember, 39 to 41,000 years ago. This one also produced a genome. And this, you'll never be able to see out. I'm going to zoom in on a second. This shows 22 chromosomes of genome data from this incredibly decently preserved piece of bone that was analyzed in Leipzig. And you can see that if we look up closely, you can see that once again here on this particular chromosome, you can see we've got individuals from Africa at the bottom. The D is the Dinka That's a sub-Saharan African person. French, and then a Han Chinese, and then our friend Yu, which is Ustashim. Can you see that one here? Ustashim, this one here with the yellow. And then above that is the Awase, and where you see green, this is Neanderthal integration. So that's Neanderthal DNA in this person. And you can see that it's present in big chunks here, really big chunks. In fact, around half, maybe, or a third of this entire section of this chromosome is deriving from Neanderthals. This suggests that this person lived much closer to the integration, much closer to the gene flow period than the Ustashim man. This man probably has a Neanderthal great-great-grandparent. 4 to 6 generations before this person lived, he had a very close relative that was a Neanderthal. So this puts an incredibly tight amount of time on the integration in this particular part of the world in Romania. So now we know there was not just one integration, there were probably several. And the other really intriguing thing to point out here as well is that both of these modern humans are evolutionary dead ends. They have no relatives amongst us today. So we're still looking for those people at the moment. And more DNA data, more dating evidence is needed until we can be sure. Okay, moving rapidly on now. Denisovans. I have to talk about Denisovans. I'm really privileged to be working at the site and part of the team that's working on the chronology of this particular site. One of the most amazing sites in the world, I think. I think it's fair to say without too much hyperbole. Denisova Cave is in the Altai region, about halfway across Russia, at the point where Kazakhstan and Mongolia and China meet Russia, at this little pinch point. And the Altai region is a really fascinating place. To get there, you fly into Novosibirsk, with the biggest city in Siberia, and then you drive for about 11 hours. And eventually the steppes, the big flat endless steppes of Siberia give way to an alpine environment. And it's a really beautiful place, beautiful part of the world. You've got like big eagles soaring in the sky and lots of quaint little villages with Altayan ethnic people living in it, whose ancestors go back to include Genghis Khan and people like that. And if you look at this picture, you're looking down here at the camp of Denisova. So this is the camp down at the bottom here on the banks of the Anui River, which is near where the site was discovered. When the Russians first found the site in 1978, they had to get in there by helicopters and tramping. It was a really out-of-the-way place. There were no roads. But in true Russian style, they've created not only a road, but also a chalet-like camp that you can go and stay in, and they have a permanent excavation team, or semi-permanent excavation team there. So this is the little chalet where there's a kitchen area, and then they've beautiful facilities. It's really nice. They've even got a spa and a sauna. I mean, this is unlike any place you go if you're working in this period, but believe me. Now, the site itself is just along this river, about 250 meters away from the camp, and it consists of a large entrance hole and a smaller hole at the side. And the site has been excavated since the mid-1980s. It's several meters deep. More than 7 or 8 meters have been excavated down. And as you go into the main gallery here, this is a 3D reconstruction. On your right-hand side, down at the base of there is the main gallery. So just down where you're looking at is the main area of the site. But the key stuff is to the right of this. And as you come into the so-called East Gallery and you look down, this is about 7 or 8 meters down here, and you go down just through here where these ladders are. This is where some of the key fossil evidence has been discovered that tells us all about these interesting individuals called Denisovans. In 2008, this tiny finger bone was excavated in the East Gallery in layer 11, which is, we think, 50,000 years plus. And this is the small bone of the finger bone of an 8 or 9-year-old girl. And the phalanx is exceptionally well preserved in terms of DNA and collagen. And this is because the site of Denisova Cave has a very, very low temperature all year round. It's rather like a fridge. It's a perfect place for DNA preservation. And in fact, this finger bone had more than 70% of its DNA still in it, endogenous. Most bones that are analyzed in this period have less than 1%. Most of the DNA is bacterial and contaminating, but this bone has 70% endogenous. The bone only weighed 29 milligrams, but it produced a nuclear DNA sequence, 99.9% extracted, done dozens of times to produce the same result. It's a better genome than the Neanderthal genome that took so long to make, just because of the preservation state. So what does it tell us? So amazingly, when the DNA was analyzed, it showed that this wasn't a Neanderthal, and it wasn't a modern human. It was something else, this mysterious third hominin. And it was initially given the name hominin X. But then later it was decided to name it Denisovan, after the cave. But of course, you can't give it a formal species name on the basis of a tiny fragment like that, because you have to describe the bones when you report a new species. But intriguingly, this individual sits outside Neanderthal and modern human DNA. So this is a kind of a family tree. In the grey here, all modern humans outside and including Africa, all people. And then in the blue here at the bottom, Neanderthals. So we have a common ancestor 380 to 400,000 years ago. And then at the very bottom, this little Denisovan individual, which again shares a common ancestor a little bit before that, about 1 million years ago perhaps. So if we zoom down here, you can see Denisovans sitting just off our close cousins. So if you imagine how this might pan out, so we're talking about an individual that is as far away from a Neanderthal as we are from a Neanderthal. And intriguingly, of course, it was discovered in 2011 that the Denisovan individuals had interbred with modern humans as well. So that not only was there a Neanderthal interbreeding or gene flow event, but there's also gene flow from Denisovans into modern humans. And fascinatingly, the groups that we see in the modern world that have Denisovan DNA include people in Eastern Asia, in Melanesia, and in Australia. This is the kind of summary diagram that I'll show you before the end of my talk, which gives you a kind of a rundown of the complex nature of what we're now terming the leaky replacement model. So what you can see here are modern humans in the blue here, and Denisovans here in the middle in yellow, and Neanderthals over there. And this is a kind of a family tree of evolution. And you can see here that there are periods, and there are evidence for gene flow coming in these red arrows from Denisovan integrating DNA into Oceanians, a bit between 3 and 6%, about 0.2% into people in Asia. We've got Neanderthal integrating DNA coming into people prior to the dispersal of modern humans around the old world. We have Neanderthal DNA going into Denisovans as well. The picture is becoming extremely complicated, but nonetheless very fascinating. Last week in London, 2 weeks ago in London, we were at a meeting where they reported 3 new genomes of Denisovans, which showed once again there was interbreeding between Denisovans and Neanderthals in the same cave at Denisova through time. The hunt for fossils goes on, but it's going on in a really intriguing way. This is my PhD student, Samantha Brown, who's working in Oxford, and we're using a really interesting and revolutionary, I'd say, technique that was developed by a colleague of mine called Michael Buckley in Manchester University, and it's called ZOOMS, which means zooarchaeology by mass spectrometry. To cut a very long story short, all animal bones contain slightly different sequences of proteins and peptides in them, which correlate to species. So if you can measure these, you can get a kind of a fingerprint. So you can identify bones based on these protein sequences. So here, for example, this sequence here belongs to a human. And this one, you can see it's very different, belongs to a reindeer. And here, a hyena. Now, at Denisova Cave, 95% of the bones are unidentifiable. They're tiny fragments because they've been chewed up and eaten by animals like hyenas. And as a result, archaeologists can't identify them. No one can say, oh, look, this is a piece of a hyena, because it's too fragmented. Some of the bones can be identified, but these are the bigger ones, and they're comparatively rare. So what we've been doing is using this technique of zooms, which allows you to take a tiny amount of the bone, measure the abundance of these different peptides, and put an identification on the bone. Now, why are we doing this? Because the bone is so well preserved, if we can find even a fragment of the bone that could be a human, we can get a genome, and then we can get all this amazing information about the relatedness and the population histories of these people. So 2 months ago, after analyzing 2,513 bones, poor Sam took a month, found this. And this tiny little 2.5 centimeter bone, completely unidentifiable, we've just got the result back from Leipzig. It's a mitochondrial DNA analysis, tells us this is very highly likely to be a Neanderthal. So this tiny little bone, very well preserved, it could give us another genome, which is extremely useful information because it tells us a lot about the relationships between these people. We've dated this, and we think it's about 80,000 years old. So in summary, humans evolved in Africa, I'd say around 200,000 years ago, and then perhaps around 50 to 60,000 years ago, we made this move around the world, and we populate the world all over since then. Everyone in the world is modern Homo sapiens sapiens, but we have this intriguing evidence for genetic exchange or gene flow between different archaic populations that lived outside Africa prior to our movement out of Africa. And these include Neanderthals and Denisovans. And these little variable proportions of DNA that we get from these people come with us today, and we can now intriguingly map and identify what the function of these genes that we inherit from them are today. And this is the subject of active research. I'd like to thank the organizers for bringing me to New Zealand to give this series of lectures, the Alan Wilson Centre, and everyone here who's been involved in coming to Nelson, which has been a real privilege. I'd like to thank the Paleochron team that I work with at the University of Oxford and the European Research Council for funding all of this work. We're very grateful to them. If you want to know more about what we're doing, you can check our website at paleochron.org. We update with a blog all of the work we do. And it would be nice if you could get in touch with us if you have any more questions, maybe in the future or now if you do. Thank you very much for coming tonight, and thank you for your attention.